Hello great friends. We are going to cover terms used in genetics. Genetics is a science, which deals with the study and understanding of heredity and the variation of inherited characteristics. Variation in genetics is the range of differences between individual organisms. For example, differences in skin color. Continuous variation is a type of variation where all types of features have a normal distribution. For example, the height of individual human. Discontinuous variation is an abrupt variation in which there are few or no intermediate forms. For example, different color of flowers in a single plant species, different types of blood groups in individual human beings, and different types of earlobe. Chromosomes are the thread-like structures found within a nucleus of a cell. DNA, which stands for deoxyribonucleic acid, is the molecule that contains the genetic code of organisms. RNA, which stands for ribonucleic acid, is the molecule that acts as a messenger carrying instructions from DNA for controlling the synthesis of proteins. A gene is that portion of a DNA which codes for a protein. The total set of genes in a cell is called the genome. Allele is the variant form of a gene, controlling a particular trait. Nucleus is a double membrane-bound structure made up of a viscous fluid known as nucleoplasm in which nucleolus and chromatin materials are suspended. Haploid nucleus is a nucleus containing a single set of unpaired chromosomes, example in sperm and ova. In humans, the haploid number is 23. Diploid nucleus is a nucleus containing pairs of chromosomes, example in somatic cells. In humans, the diploid number is 46. Genotype is the genetic makeup of an organism. Phenotype is the characteristics visible in an organism, controlled by the genotype. For example, a tall plant or a dwarf plant. Homozygous is having a pair of identical alleles controlling the same characteristics. Heterozygous is having a pair of dissimilar alleles for a characteristic. Dominant gene is a gene that always shows in the phenotype of an organism whether the organism is heterozygous or homozygous. Recessive gene is a gene that only has an effect on the phenotype when the organism is homozygous. Complete dominance is when a dominant gene is seen in the phenotype of an organism and the recessive gene is not. Incomplete dominance is when one gene does not completely dominate the phenotype of an organism and a blending between the two traits occurs. Example, red flower gene and a white flower gene combine to create a pink flower. Codominance is where heterozygous individual expresses both simultaneously without blending. A TESTCROSS is a valuable way to use a genetic cross to determine the genotype of an organism that shows a dominant phenotype but unknown genotype. Multiple allelism is a condition in which a gene exists with more than two alleles for a given trait, and any two of the alleles may occupy the same gene position on homologous chromosomes. An example is on the gene determining blood groups. Linkage is the process whereby genes are located on the same chromosomes. Haemophilia is a genetic disease due to absence in red blood cells of clotting factor 7, and this is because of the absence of the gene which codes for this factor. Muscular dystrophy is a genetic defect due to recessive gene characterized by muscular wasting. Mutation is a sudden change in the genetic constitution of an organism caused by factors called mutagen. Chromosomal mutations involve sudden changes in the structure of chromosomes. Gene mutation refers to the change in chemical nature of the gene. Sickle cell anemia occurs due to formation of abnormal hemoglobin known as hemoglobin S. Albinism is a condition due to a recessive gene which leads to lack of synthesis of the dark skin pigment termed melanin. Goodbye friends. Thank you for watching. Remember to like, share and subscribe to our channel.